So, hello everyone, welcome back to my YouTube channel. I am Nandini and I scored 632 out of 720 in NEET 2024 examination, and currently I'm preparing for NEET 2025 because obviously this year scam and all happened, and that's why I could not get a government MBBS seat. So, let's see, hoping for the best next year. So now I was studying photosynthesis and respiration. So I just thought, why not to share with you all my trick to remember the cycles and the all of the glycolysis and Krebs cycle and reaction and C3C4 cycle that helped me to not commit any mistake regarding these two chapters in my botany section of NEET 2024. For your all information, I scored 180 in zoology and 165 in botany. So those were the questions from, I think, genetics, and one was from I even do not remember now. So just let's keep it aside. Okay, so without any further wastage of time, let's get into the video. And I can just guarantee you this, that once you are completed with this video, you will not face problem in further times if you watch this video till the end. And do let me know how did you find the video. So this is my NCERT of biology, class 11th, photosynthesis. Okay, let's start with. Okay, so the first cycle which we come across is C3 cycle. So these are my kind of cheat notes which my teacher provided me. So it really becomes very easy to learn the cycle from this. So you all can also take a screenshot and learn from this. I hope that you are getting a wider view. Okay, so this is the dark reaction, Blackman's reaction, Calvin cycle, C3 cycle, biosynthetic phase, carbon assimilation, photosynthetic carbon reduction, reductive pentose phosphate. So these are all the names credited to C3 cycle, which was discovered by Melvin Calvin. So this is the cycle. First of all, we have taken it for 1 mole of glucose, because 1 mole of glucose is C6H12O6. Right, this is the formula for glucose, and it contains 6 carbon, so 6 CO2 is being fixed by the enzyme Rubisco, which is ribulose 1,5-bisphosphate carboxylase oxygenase. Okay, and RUBP is the 5 carbon compound, and we are taking for 6 mole because we are making 1 mole of glucose. Okay, so we are taking it for 1 mole of glucose. So RUBP is the primary substrate to which CO2 is added in the presence of the enzyme of Rubisco, which is present in the stroma and matrix, and this makes a 6 carbon compound, which is the first product of C3 cycle, and this is unstable. 6 molecule of H2O is being added to this compound, which makes 3-PGA, which is the stable product of C3 cycle. That is a 3 carbon compound, and that is why the name C3. Okay, now this is like 12 moles. This is 3 carbon, total of 36 carbon. Fine. 3-PGA now gets converted into 1,3-bis PGA, then 3-PGAL, and then here is the reduction phase. This is the carboxylation phase, and this is the regeneration phase. You all know that the C3 cycle is divided into 3 subphases, which is carboxylation, reduction, and regeneration. So this 3-PGA is converted into 1,3-bis PGA. So here obviously 1 phosphorus is being added, which means that 1 phosphorus is needed. So it comes from the ATP, and since 12 mole are there, so 12 ATP are needed, and that is converted into 12 ADP. Now this 1,3-bis PGA is further converted into 3-PGAL, and here 12 NADPH2 are required. So this is converted into NADP plus. Okay, now this is 12 mole, and again 3 carbon, because nowhere CO2 has been removed. So this still remains 36 carbon total. Now this 3-PGAL is converted in 2 moles of this 3-PGAL is taken for the process, and 10 moles are taken for the regeneration of RUBP. Okay, so 12 moles were here, and now 2 moles are taken over here, and so 10 moles remain, and these 10 moles are again converted into RUBP. 6 ATP is required over here. Okay, so this is the step of regeneration. So this is the whole of the Calvin cycle. I think that it is clearly visible. So this was the first cycle. I hope you got it. Now moving to the second one. I'll try to keep the video as short and as crisp as possible. Okay, so there are certain questions also at times. See, the further what happened is that this 3-PGAL is taken for the further processes to make 1 mole of glucose. Fine. So immediate H donor is NADPH2. I think that this is now visible. This is the NCRT wala thing, but as I said, that my teacher provided me with this, and this is very easy to remember, so you can go with this. Now coming on to the Hatch and Slack pathway. Hatch and Slack pathway is not that given detailed in our NCERT, but you can say that the C4 pathway is given, which is also known as Hatch and Slack pathway, cooperative photosynthesis, dicarboxylic acid or CO2 concentrating mechanism. That is the C4 pathway. So it is an advanced mechanism, we all know that. So I'll be briefing you over here that what all is being happening. So this is the bundle sheath cell, and this is the mesophyll cell. Here the actual Calvin cycle occurs in the bundle sheath cell, and here 12 NADPH2 and 18 ATP is used for 1 mole of glucose molecule. And here the primary substrate is phosphoenol pyruvate. Here you can see it, phosphoenol pyruvate, and then a C4 acid, which is oxaloacetic acid, which is the first stable product of CO2 fixation in the C4 plant, is made, and then the further process continues. Fine. And here the pepcase is used, and here 12 ATP are used, and for this 12 NADPH2 and 18 ATP are used for Calvin cycle that occurs in the bundle sheath cell, and in mesophyll cell the primary fixation occurs. I hope it's clear. Moving on to the third cycle rather. This is the C2 cycle, which is also a wasteful process, which we all know that it is photorespiration, and also known as photosynthetic carbon oxidation cycle, PCO cycle, glycolate mechanism. And this occur in 3 different organelles, which is chloroplast, peroxisome and mitochondria. This is not given in as detailed in NCRT as it has been described here, but you can take a screenshot for your convenience if you want to study it in detail, but not much detail is required, but you can for once view C2 cycles. Okay. Now, so all the cycles of photosynthesis are over. Let's jump into the respiration process. So respiration, the first very important, which occurs in all organisms, whether be it eukaryotic or prokaryotic, which is glycolysis, which is a fundamental universal reaction occurring in all the organisms which are living except for viruses. So I'll help you remember this because this is very easy. I remember it with a trick. Okay, so the most important things which are there, I'll be highlighting those for you. Hope it helps you. So glucose is converted to glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, then it converts into DHAP and 3-PGAL. So these two are interconvertible with the help of enzyme isomerase. That DHAP also gets converted into 3-PGAL, 3-PGAL to 1,3-bis-PGA, 3-PGA to PGA, to PEP, which is phosphoenol pyruvic acid, then further to pyruvic acid. So like once or twice if you will write it now, then you'll be able to remember it very easily. So the major enzymes included are hexokinase, phosphofructokinase, and then here pyruvic acid kinase. These are the important enzymes I'm telling you all about. You can also take a screenshot for this. This is a very easy way to remember. Okay, and yeah, I think that, see, this is the PAO phase, this is the investment phase, and what all irreversible reactions, 1st, 3rd and 10th, and the pacemaker enzyme is the phosphofructokinase, which is used in the 3rd step of glycolysis, and this occurs in the cytoplasm, and it is also known as EMP pathway, Emiden-Meyerhoff-Parnas pathway. So I hope that this is now clear to you all. Now moving on to the second one, that is Krebs cycle. Again, one of the nightmares for NEET aspirants to remember this huge cycle. I know it's kind of a bit tricky, but we have not got any other options beside remembering this. So let's just see now what happens. So 1 mole of pyruvic acid, which was made in the glycolysis, is converted into acetyl coenzyme A, which is the link reaction. Okay, so this is the link reaction, and these are O2 dependent but not O2 involved steps. These are all the key points that are very much frequently asked in the test series, which you should keep in mind. So I'm just highlighting these for you all so that you do not face any problem while giving your tests. So here, link reaction, which is the conversion of pyruvic acid into acetyl coenzyme A, because pyruvic acid enters the mitochondrial matrix where this reaction occurs. So here Mg2 plus is a very important factor for this. Okay, and here first decarboxylation occurs. So pyruvic acid enters and it is converted into acetyl coenzyme A, which is a 2-carbon compound, and pyruvic acid is a 3-carbon compound, which means that CO2 needs to be removed. And fatty acid also enters, also is converted into acetyl coenzyme A by beta oxidation. So beta oxidation of fatty acids takes place to get converted into acetyl coenzyme A. Now this process continues, and then the cycle continues, which is known as the Krebs cycle. Okay, so what happens is that acetyl coenzyme A is converted into citrate. So cis aconitase, isocitrate, oxalosuccinate, alpha ketoglutarate, succinyl coenzyme A, succinate, fumarate, malate, oxaloacetate. A bit tricky to remember, but you can take a screenshot and just revise this once or thrice. You'll write it, then you'll get the process. Okay, so each turn of tricarboxylic acid cycle, which is generates 3 NADH2, 1 FADH2, and 1 ATP or GTP. So I think that this is clear to you all. Now let's get into the next cycle. Okay, so I think that all the cycles involved in the chapter are over. So yeah, here once I wrote why give up, no won't ever. So yes, keeping this in mind, we all will be fighting for NEET 2025 and we will all come out with flying colors. So if you found this video helpful, then do like, share and subscribe. It really motivates me to make further videos. And if it helped you, then do comment, and I'll really be glad to hear your comments. Meet you in the next video. Till then, keep hustling, keep studying, and thank you.